Hello dear students, I am Dr. Sharad Kumar, Assistant Professor, Department of Wildlife Sciences, Aligarh Muslim University, Aligarh. Mammals play a very crucial role in biodiversity maintenance. Therefore, estimation of their diversity and abundance is necessary for planning of biodiversity conservation in a particular area. In this lecture, I shall discuss about various methods used to estimate the diversity and abundance of mammals. Need of monitoring of mammalian diversity. Trends in species diversity are used to assess human impacts on animals and to identify threatened populations or species or subspecies and important areas for the conservation of mammals. Diversity estimation is also essential to monitor change in diversity over time or among habitat types and designing the management strategies for the conservation of mammals. In addition to this, monitoring of mammalian diversity is necessary to evaluate the success or efficacy of wildlife management programs from time to time. Methods to estimate mammalian biodiversity. There are various methods to determine the diversity and abundance of mammals, and there is no universal method which can be applied to all species or applied in all conditions. Relevance and application of a particular technique or method to a species depend on behavior of species, habitat and local conditions, and the research questions. Selection of one technique or methods over another depend upon the following factors. How data are to be used, or what is the objective, and time, manpower, equipment and cost constraints of different methodologies. The method that can be used to determine the diversity and abundance of mammals can be divided into two categories: direct methods and indirect methods. Direct methods. In these methods, we collect data or information on species of interest through the direct sighting of animals, for example, live traps, block count, transit, camera trapping. Indirect methods. In these methods, we collect data or information on species of interest by having observation on the signs left by the animals, for example, pellet count, dung count, track count and acoustic surveys. Let us understand these methods. First, I shall go through the direct methods and then through indirect method of sampling of mammals. Live traps. Live traps can be used to estimate small and medium-sized mammal species richness, composition and abundance across ecological gradients. In the usual trap design, a lever or treadle is pressed by animal which shuts the door behind an animal caught in the trap. Various traps designed to trap different animals are available in the market and they can be used as per the need of the survey. Salmon traps available in different size for the small mammals and can be placed in the habitat of small mammals to trap them. In addition to this, people also use pitfall traps in the areas which can be dug easily. Pitfall trap consists of a container buried in ground so that its top is level with the surface of the ground. Any creature that wander nearby may fall in it and trapped in the pitfall trap. For arboreal mammals, the traps can be tied on the thick branches of the trees used by the arboreal species of mammals. For aquatic species, traps should be placed on rafts in water which are then anchored or can be placed along the water edge. Trap must be positioned in aquatic habitat in such a manner that water should not enter even after rain or high tide. Traps should be marked to locate them easily on the successive inspections. In dense habitat, nylon line can be used to locate the traps. To set up traps effectively, it is crucial to have the knowledge of behavior of the focal species. Setup of traps. Let's understand how to set up the traps in the habitat of the mammals. Divide your target or study area into square grids and set up the sufficient number of traps in each grids or in the selected grids. Spacing between the traps will depend on the distance moved by the individuals of a species, for example, 5 to 20 meter for small mammals. Trap location will be marked with cane to locate them easily in the field. Baits can also used to attract the animals to traps. Weight material should be dry and will not decay. This method provides you the information on the species diversity and abundance of small mammals in the area. Number of captures divided by the number of trapping nights or the trapping days give us index of abundance of mammalian species. If the method is combined with mark, release, recapture method, then we can determine population size of mammalian species caught in the traps. Block or total count. The simplest way to determine how many individuals of a particular species of mammal live in a particular area is to count all of them. This method is suitable and effective for the conspicuous large mammals. To use this method, divide the area into blocks of suitable size or variable size based on the natural features. Count the number of individuals present in each block on same day and same time. So we need one team of surveyors in each block. To use this method effectively, research teams should have the fair idea of map of the area and various natural features present in the area which can be used to divide the area into the blocks. Camera trapping. Camera trapping has become an increasingly popular method to determine the diversity and abundance of mammals and biodiversity values. Automatic camera traps can collect data on wide range of species, which is important with the current demand for multi-species monitoring. It can determine the mammalian species presence, abundance and population change, particularly in the face of deforestation and habitat destruction. The method enables collection of baseline population data on elusive mammalian species. The details of this method will be discussed in the separate lecture. Line transit. The line transit method involves traveling or walking along a straight line laid in habitats of mammalian species, recording the individual on each side of the line. The transit should be laid randomly in the habitat of the species. To lay the transit, a baseline is drawn across the survey area such that perpendicular lines from it would cover the entire area, and these perpendicular lines become the transit to collect the data. The baseline is then divided into widths the same as the transit width and walked by 2 to 3 persons in the morning and evening hours when animals are more active. Whenever an animal is sighted, data on species, number of individuals, sighting angle, which is the angle of animal from line, and sighting distance, which is the distance of animal from the observer. This figure provides a systematic view of laying of line transects to determine the abundance of mammals. This figure is providing illustration of basic measurement that can be taken for individual sited along the line transects. L is indicating the line and X is an animal sited on the side of the line transect. Ri is the sighting distance and OI is the sighting angle. These two measurements are used to calculate the perpendicular distance yi, which is used to calculate the density of mammalian species present in the surveyed area. Assumptions. Following are the critical assumptions of line transect methods. Assumptions. Following are the critical assumptions of line transect methods. Animals directly on the line are never missed and must be recorded by the observer. Animals are fixed at the initial sightings positions. They do not move before they are detected by the observer. Distance and angles of sighted animals are measured exactly with no measurement error and no rounding of the measurements. Sighting of individual animals are independent events. Animals being sampled are distributed at random. To meet the last assumptions, transects are laid randomly in the different habitats of the animals. If these assumptions are valid, we can estimate the density of the animal using the equation D is equal to N upon 2LW, where D is equal to density of animal per unit area, N is equal to number of animals seen on transects, L total length of transect, W width of the step or the width of the transect. But now computer softwares like distance are available to analyze the data we collect on the transect, and we can use computer software for the analysis of the transect data to calculate the density of the different mammalian species recorded on the transects. Indirect methods. Counting dungs. For some species, dung is more conspicuous in the field and easy to locate. For example, we can easily locate the dung of elephant, rhino, gaur, wild buffalo in the field. For such species, dung count is a reliable method to determine the presence and abundance of a species in a particular area. If dungs are abundant, quadrates can be used for counting the dungs. Otherwise, transects can be used to count the dung in the field. Length of persistence of dungs varies according to habitat and weather conditions, dung composition, fiber content and abundance of dung beetles. So, there are two approaches to deal with it. First one is clear dung from marked areas and return before any fresh dung has had time to decay and count the dung deposit in the interval. The second approach is mark some very fresh dung initially and on revisit use these marked dungs to compare with dung in the field and ignore any dung that looks more decayed than the fresh one marked at the first time. This method provides information on presence and relative density in different areas. By assessing the amount of dung produced per day from either field observation or studies on captive animals, the dung densities can be converted into number of mammals present in a particular area. If using defecation rate from captive animals, remember that Captive animals have high quality diets and have different defecation rate than wild individual of the same species. In addition to this, dung of different age and sexes may differ in size and shape. So we should take into consideration all these points while counting the dungs in the field. Pellet group count. This method can be used to determine the relative abundance and population density of mammals, especially ungulates. Pellet group is defined as cluster of 6 or more pellets produced at the same defecation by an individual. Two groups lying close together or one on top of the other can usually be distinguished by difference in color, size and texture. Pellets groups can be counted in 10-meter circular plots or 10 into 10-meter circular plots or some smaller plots. Species and number of pellet group of each species are counted. In case of groups found on the edge of a transect or plot, 50% groups are included in counting and rest are excluded. Line or string of pellets is considered one pellet group and generally made by moving animals. Number of pellet group per unit area provide the estimate of relative abundance of mammal in different areas. Estimate of population density and size from pellet count. Pellet group count to estimate population density fall into two main categories. The first one is fecal accumulation rate or clearance count, and the second one is the fecal standing crop counts. Both methods use information on fecal pellet group decay and defecation rate of different species present in a particular area. Overall accuracy of the population estimate will be influenced by accuracy associated with the estimate of defecation and decay rates as well as the sampling protocols. Clearance count. FAR is most suitable in site of high ungulate density, means areas having more than 30 individuals per square kilometer. Area of interest is divided in various habitats and sample plots should be allocated to each habitat. Plots can be on transects or without transects and clearly marked on map. Marked with clear visible mark to locate them easily on the subsequent visits. Plots up to 7 into 7 meter in size if it will be searched by one person, and 10 into 10 meter plots if it will be searched by two people are used to count the pellet. Dividing the plot into 1 meter strip which are systematically searched is the most practicable searching technique to count the pellets effectively. If two people work together, they should start at diagonally opposite corners and both search the whole plot. This will provide you the accurate counting of the pellets in a particular plot. Search and throw out all the pellets or dung from the plot. Number of groups found during this initial search may also be recorded to allow a population estimate based on standing crop data. Fresh pellet group found within or close to the plot should be clearly marked to enable any decay between visits to be estimated or to be conducted to determine the pellet density in the plot. All plots should be revisited a second time after the appropriate time interval. Number of pellet groups accumulated since the first visits are counted. Where previous pallet decay data are available, the second visit should be just before the expected decay time. If no decay information is available, a period of 2 to 3 months is usually acceptable for counting the pallet after clearance of plot. On the second visit, the marked groups should be examined to determine whether there has been any loss of pallet group due to decay between the two visits. If so, then the figure for accumulated fecal pallet groups should be adjusted to take account of the decay. By using this formula, you can calculate the density of the animals. Number of ungulates per hectare is equal to number of pallets group divided by time between visits in days multiplied by defecation rate. Standing crop plot counts. This approach is best suited to areas of medium ungulate density between 10 to 30 individuals per square kilometer. First, stratified the area of interest as per habitat, and then plats should be allocated proportionally to the various habitat. Count the number of pallet groups or dunks of different species in appropriate size of plots and calculate the density of species by following data. Number of ungulates or deer per hectare is equal to number of pallets group per hectare divided by average decay time multiplied by defecation rate. Counting footprints, runway and pug marks. Tracks of species in soft ground or snow are an excellent way of determining presence and absence and relative abundance of the species. Footprints can be assessed actively or passively in the target areas. Active assessment typically use scents or weights to bring animal into the tracking plots, while passive assessment use tracks made by animal in their daily travels. Set out plots large enough to provide good tracks, to rake them, clean and revisit them at the fixed interval and record all the tracks present in the plot. For snow tracking, the intervals are usually set by fresh snowfalls. The important point to remember is that old tracks are not confounded with the new tracks. A similar kind of approach can be used for small mammals that make runways in grasslands or the woodland habitats. These are the pug marks of male and female tiger. Initially, pug mark census is widely used method to determine the status and distribution of tiger in India, but the method is now replaced by the camera trapping. Acoustic surveys, generally used for bats and marine mammals, as visual surveys has some limitations. Bats and other mammals can be monitored by recording the vocalizations. Advanced equipment are now available to record the vocalizations of mammals. Based on recorded vocalizations, we can identify the species of mammals or to some extent the individual of a species of mammals. Recording and identification of vocalization help in monitoring of bats and marine mammals in different areas. Acoustic recorders and hydrophones are used to record the vocalizations. Computer softwares are used to analyze the recorded sounds, and based on these sounds, the scientists provide the indication of the diversity and the abundance of the bats and the marine mammals. Thank you.